All right. So you see this, everything I'll be explaining now, first is just to reiterate some of the things I've talked about already. Um, this is just showing you the linear. So the light reaction is linear flow. It's just showing you the linear flow of electrons as it flows from P680, which is PS2, goes to the primary electron acceptor and then transfer through the line up till the point that it's going to get to the P700. Then the electron from P700 goes to electron acceptor, NADP plus. So there are 8 steps in the linear electron flow. A photon, like I showed you here, a photon is the light harvesting complex or antenna complex. Antenna in that it harvests harvests sunlight, or it's able to pick sunlight. So light harvesting complex of PS2. PS2 comes first, don't forget again. And you see its energy is passed among the pigment molecule until it's going to get to P680, which is the chlorophyll A, the reaction center there. An excited electron from P680 will be transferred to the primary electron acceptor here, which refers to the oxidized form of P680 plus. So P680 will be oxidized, right? And now it's going to be P680 plus because it's oxidized. Now, next thing is an enzyme will catalyze the split of H2O to form into 2 electrons, like I told you, like I analyzed for you there, 2 electrons, 2 hydrogen ions, and oxygen molecule. The electrons are transferred to the P680 that was excited, that was oxidized, so that it can reduce it back to its original stable state, which is now going to be P680 and not P680 plus again. The H plus are released into the thalocode space. The oxygen atom combines with another oxygen atom generated by splitting of 2 water molecules, which is why I did 2 the right time, right? To form O2. Electrons are then passed through the series of redox reaction from the primary electron acceptor here, and it goes down from PS2 through the transport chain to PS1. The electron transport chain include PQ, which is plastoquinone. It includes cytochrome complex and plastocyanine, PC. Energy released by electron transfer is used to pump hydrogen ion into the thalocode space, creating a photon gradient across the thalocode membrane. Now, the potential energy that was stored in the proton gradient drives the production of ATP, which is what created ATP. Think about chemiosmosis in cellular respiration. That's the same way that it happened here because it doesn't require phosphate, right? Now, in PS1, transferred light energy in PS1, we excite P700, just like it excited P680 as well. Now, don't ever mix it up. PS2 have the smaller wavelength, P680, meaning that lower energy, higher wavelength, right? So it has P680, whereas PS1 have P700. So in PS1, the transfer light energy excites P700 as well. And then it's going to lose electron and become excited to the primary electron acceptor, which will later transfer it to NADP plus. Now, P700 that was excited is now missing an electron, so it becomes P700 plus. It's going to accept electron that was passed down from PS2 that comes this way, and that will make it become stable. Electrons are passed down from primary electron acceptor of pH1 down a second electron transport chain to protein ferrodizine. Ferrodizine, there's no proton gradient or ATP produced by this electron transport chain. The enzyme NADP plus is going to cause NADP plus. Enzyme NADP plus reductase will reduce NADP plus by giving it the electrons and reduce it to NADPH. 2 electrons are needed to reduce NADP plus to NADPH, which is the 2 also that was lost. The electron of NADPH are at higher energy level than they were in H2O, so are more readily available for reaction at the Calvin cycle. Do you know what that means? The electron that was released from here, because these are the later end of the cycle, right? Of the phase of the linear chain rather. So the electron here, they still have higher energy, because the more they travel, the less energetic they become. But these guys are still fresh. Their energy is still fresh. So they are able to power more they have more energy concealed in them that they can take to the to the Calvin cycle than the electrons that was in the water molecule. That's what that that line was explaining. The formation of NADPH also removed an H plus from the stroma. Now, this is the chain that we have just explained from number 1 to H. And I believe it's clear, so I will go ahead and not run through it again. Now, let's look at this mechanical analogy of light reaction. Light reaction use solar power to generate ATP and NADPH by providing the chemical energy and reducing power needed by Calvin cycle to make sugar. The energy changes of electrons during linear flow through the light reactions can be shown in a mechanical analogy. So this is more like what we are talking about. The you see photosystem 2, photosystem 1. The electron comes in, photon release, electron is released and then captured by the electron acceptor. The electron acceptor is here, then drive the electron through the electron transport chain. It goes that way, comes here. This guy also comes here, split this releases electron, the electron becomes excited. Then the electron coming from here is going to balance out the electron that was lost here. So the photosystem becomes stable again. And then the electron that was released by photosystem 1 will later be accepted by the NADPH, which will be reduced, which is powered by NAD plus reductase. All right, so that's all that happens there, just to give you a picture. Now, this is cyclic portion of the stage. So I think for time's sake, let me stop this here. Then I will do the dark reaction or light independent reaction in a separate video that I'll upload later. Oops. Okay. I'm not going to record